Well, welcome. We all know that biotech is a driver in our Massachusetts economy. Tonight we're going to explore why somebody would start a biotech company or a tool company or a company in general as a startup. We have two guests here that have agreed to walk us through what they started, how they started it, and more importantly, why they started it. So why don't we start with introductions?
Sure. I'm Liz Reczek. I'm the CEO of SeqLL. Just to give you some history on my background, I have a cancer biology background. I did a PhD in cancer biology at MIT and then a postdoc at Harvard Medical School. And then decided to join a small startup doing antibody combination therapeutics. That was called SeqLL, and I joined there early on as a research scientist and helped develop a technology for discovering fully human antibodies from human patient samples. And then for expressing them as mixtures. And the goal there was really to generate mixtures of antibodies that could be given to patients that were suffering from infectious disease and to treat them if they weren't able to fight off that disease themselves. And that's where I really got the bug for being in biotech and in the startup environment especially because of the way that the teamwork that environment was so, teamwork was so important to really building a technology and creating a successful company, and I really enjoyed that. Through my experience there, I worked my way up and worked at various roles from research scientist to director of research, and finally ended up leading that company as the CEO. And went through all the different stages of that company, from the heady days of good funding and good research to finally our investor pulling out and then having to go out searching for other sources of funding and partnerships, and eventually watching that company crash and burn, unfortunately. But that didn't diminish my excitement for the biotech space, and I looked for other opportunities after that to get into the startup environment again and get back to that building that I really enjoyed and teamwork-oriented environment that I really enjoyed.
Well, building on what Liz said, it's the reason why we're here. 80% or more of startups will fail, and we've all gone through it. And that's what brought us to this point in our lives where we're working together to keep a promising technology alive and to bring that out into the world. And so as far as an introduction, my name is Daniel Jones. I'm one of the founders of SeqLL. We founded the company back in 2013, but before then I grew up in Massachusetts, in Drake. I attended St. John's Prep, then Trinity College, and at Trinity I was exposed to a lot of different types of sciences. We did capsaicin research. I did research on the Enigma machine. It's where the catalytic converter was invented. So there's some interesting background there that just opened my eyes a little bit to what was available. And I knew I wanted to work in the biotech industry in some capacity. So after college I moved up to the Boston area, started working at Exact Sciences, where it's a colorectal cancer screening assay that's now FDA approved. And like Liz mentioned, for SeqLL, went through the full life cycle. Startup, it was very successful, and that just exposed me to a breadth of molecular capabilities. They had a lot of different technologies there, and that excited me in terms of business and combining business with science, seeing what's capable to help people going forward. From there, I moved to a company called US Genomics, was there for a few years working in the research department in the wet lab, and then moved to a company called Helicos Biosciences. Worked there for about 7 years, and that ultimately led us to the company where we are today, because that company also went through the full life cycle of startup, IPO, doing quite well, and they developed this amazing technology that we've endeavored to keep alive. I'm sure we'll get into it more, but that's enough about me.
Well, that was $200 million worth of funding.
It was quite a bit of funding went through. Not sure on the exact numbers, but yes, it's very expensive, as we all know, to keep a company afloat. There's a lot of needs that you don't, I'll speak for myself, that I don't necessarily anticipate that arise as we're running the operation day to day. But they were starting from scratch. That company had nothing. There was an idea in a paper that was published from a lab in California in 2003 that spawned this company. And the venture capitalists and early scientists and other inventors, they came in and did an amazing job building this technology from scratch. It's hardware, software, reagents, consumables, even specialized molecular or specialized molecules, I should say, to perform the sequencing.
Tell us about the technology. What does it do?
Well, at its simplest level, basically it reads the DNA sequence of a molecule, of a sample. So right now, the most direct and understandable application is in cancer detection and non-invasive prenatal testing. So there's blood draws that occur at a doctor's office or really any part of a hospital or wherever. And from those blood samples, you can isolate small fragmented pieces of DNA. So everyone thinks of a human genome as the building block. And
Everybody thinks it's done, right? We map the genome. Now we know everything there is.
That's right. And what we're seeing now is, well, we know the base sequences. We know what most of us have in common. We're trying to diagnose and understand what's in a cancer pathway or what's specific to each individual. So really we're entering into this personalized medicine era, which I think my Boston accent came out there. It was personalized medicine era. But basically, we have this precision medicine initiative that's been launched by the NIH and President Obama, and I believe that that's where we fit in very, very nicely, and that's going to allow us to identify treatments, specialized treatments for specific types of cancer, specialized treatments for early detection of maybe learning disabilities or other types of autism across the spectrum. So there's a lot of really amazing capabilities that we're hoping to tap in the near future.
Well, I mean, one of the applications you hear a lot about is NIPT. We're all familiar with amnio.
Yes.
And many parents-to-be run into the trauma that, frankly, I ran into, where your physician comes to you and says there's a 1 in 300 or a 1 in 1,000 chance of having a Down syndrome baby. And the only way back in the ancient days to test was to harpoon the mother, take out some amniotic fluid, and they said there's an equal chance of a spontaneous miscarriage. No matter how small the needle seems, you are puncturing, the sac. So you're sent home with this gut-wrenching decision. Do I want to know? And do I want to risk my child so I know?
Right.
Now we're hearing this non-invasive prenatal testing. Well, why don't you explain it and the benefits thereof?
Well, for the real benefits are, as I alluded to before, you're able to avoid this invasiveness by performing a blood draw and isolating material from the mother that has negative effect or a zero effect on the fetus. So you're not causing any concern. It's just a normal standard procedure that can be analyzed and processed in a regular lab. Really for our technology, it fits right in because we're a short read sequencing platform, and that really enables us to quickly and easily identify these pieces of small DNA. And there's a fraction of that DNA in the mother's blood that belongs to the child. So as the child develops and the embryo continues to grow, the fraction that's fetal DNA increases as a percentage of the overall blood. So you're able to detect that a little easier down the road.
But you're still talking, what, 1 to 5% fetal DNA versus the total?
The range right now, it's the earlier the lower, of course, but the normal range is about 4% up to 20%. And at around 10, 11 weeks, when the initial screening's normally done, there's about 10, 11%, I believe, as the average.
Yeah, I think that's correct.
So there you're able to detect, and you're not talking major differences, right?
Major differences in?
In the DNA.
No, in this application, it's really a chromosome counting exercise. And it's made very difficult by the fact that you're looking at mostly normal maternal DNA. So the mother has a normal chromosome count, and you're looking for the fetal chromosome count, and there will be one extra copy of one of those chromosomes if the baby is, for instance, a trisomy 21 or Down syndrome baby. and you're looking for a small increase in the total DNA,
Number of 21s.
the number of 21s. And so that requires a very accurate molecule counting technology in order to detect that with a degree of accuracy that's required for a test like this, where you want to be able to give that mother very good confidence in the result that you're handing down to them, to say, yes, your baby is affected by this, no, your baby is not, and not have to go ahead with something more invasive to get that answer.
So you're able to look at 3 different counts, the 13, the 18, the 21, and different syndromes associated with each, give a good result, something that you have confidence in, and you get to save rabbits. Right? You get to do, for all of us that are very old, remember the rabbit test.
Indirect.
Indirect.
benefit
Now,
there.
I mean, now with the sticks, I guess the rabbits have been saved for many decades,
That's right.
but you're able to detect not just Down syndrome, but many other different, and I won't even say the word because I never say it right,
Aneuploidies.
aneuploidies, without putting any risk to the fetus.
Well, it gets back to sort of your original question of the risk, and we're able to detect across these different chromosomes because we know the reference of the human genome. So you know what to expect in general, and you're able to look for discrepancies. So there's a percentage of that fetal DNA in the blood that you're able to see if it skews your results one way or the other. And there's a few other approaches, but our general approach is as simple as possible. And just at the core of our company is single molecule sequencing. That enables us to have a very simple, straightforward, and unbiased sample prep. It's also low cost. So all of these wonderful components can be combined and added into this application. And that's why we really believe strongly in it. And it's one of the many examples of what we hope to do in the future.
But now, when you say single molecule sequencing, to people involved, oh yes, of course. To 5 out of our 6 viewers, they're going to look and say, what is that? How do I sequence a gene? They can understand that whenever you go to a doctor's office, they stick a needle in you, they draw blood. Depending on the color of the tube, they spin it, you get plasma, you get serum. Now what do? How would you explain to a layman how you determine the sequence?
Well, right now we're using a camera to film this show. And in our instrumentation, we use a camera to essentially film the DNA. So at a single molecule resolution, what that means is there's a high degree of magnification that is required for us to really zoom in. Because if you're looking at a sample, what we would load onto our instrument, it essentially looks no different than this bottle of water or what's inside the bottle there. It's very clear, you're not going to notice. So at a single molecule level, we zoom in and we use fluorescent tags, essentially. We modify nucleotides to attach what we call a cleavable linker. But essentially what it does is it makes the surface of what we're imaging look like the stars in the sky. So you have a very dark background where there's no objects, and then a bright signal where there's a star, or in this case, it's converted to a DNA molecule or a DNA-specific C, T, A, or G sequence. And that's performed again and again in a cyclical nature. And so if you perform those cycles long enough, you're able to extend a read from, you start with one base, and then at the end of our sequencing runs, you'll have an average read length of around 30 to 35 bases, and you're able to then extract that information, apply it to the human genome reference files, and determine what's actually in that sample.
I think it's hard for people to grasp. I mean, it was always hard to figure out on a protein. You've got, a couple dozen amino acids that you're going to mix together.
Right.
But on the nucleotides, you've got 4. Everything done in the human body is because of a combination of 4 nucleotides.
Well, that's analogous to everything done in a computer is through a combination of zeros and ones.
And ones, exactly.
So
So
it's just the order that you put those together.
So you got 3 billion slots, and somewhere in that 3 billion slot matrix, you get your DNA, and you line up C, G, A, or T in a distinct order, and that tells the cell mechanism, I want you to make this protein, I want you to make that. So essentially, you're carving that 3 billion into bite-sized pieces, putting it down on a plate,
Right.
and then since they're swans, they mate for life. The T's will only deal with the A's, the G's will only deal with the C's. You add them one at a time. But it's amazing because you got how many molecules down on a plate? 1 billion?
Billions, yes.
Billions.
There are billions of molecules that we use.
And I'm going to add in one of the 4, see which of those billions it sticks to. It's got a light bulb on it. I'm going to take a picture of it,
Yes.
take the light bulbs off, then add the next one in. Take a picture of it, and you get how many pictures while I'm building this sequence?
Well, for each cycle, we typically take almost 30,000 pictures. And then there's over 100 cycles that we perform on a normal basis. And we're working on enhancing the technology to extend that out to an extent. For different applications, like the NIPT and these cancer detection applications, you don't need to extend the runs, really.
But you're talking about millions of pictures from this camera that's got a resolution of 1 molecule.
That's an amazing feat of computation and image analysis that has to go into each and every one of these runs.
Well, I think the thing that struck me is, number 1, the resolution. You know, we talk about our cameras, we're so proud.
Right.
But a cell is this big, and you're going to divide it 1 billion times, or billions, and you're going to pick each one. Every time you add a nucleotide, take a picture. Then you end up with millions of pictures. You know, and you can think the old Xerox commercial with the monk, with the little picture trying to lay one on top of another.
Right.
But the bioinformatics programs then have to match up all these pictures.
That's right. Essentially, you're taking this picture at the same position in the XY coordinates on the stage. So we have a very high precision stage that moves this. Basically, it's a large microscope, and we're imaging the slide with a specimen on it. So it looks a little different than that. We have some improvements that make it more user-friendly than adding a cover slip in oil. But basically what we're doing is we're moving around across that slide and imaging at the exact location. We do that over 100 times for each position. And like I had mentioned a little bit earlier, there's almost 30,000 images that we standardly take. So that's quite a number. That's 3 million images total. And if you have 50,000 or so strands that are produced from that, you're talking billions and billions of molecules. And that's what you need, and that's what's so great about the technology. We're able to take your genome or Liz's genome, my genome, and process samples and have a clear picture of what's happening very quickly, in a few days even. And as far as the imaging processing goes, what really happens is that you take these pictures at the same position, you stack them up, and then you basically draw a line through them and look at every set of pixels that creates an individual molecule. So each spot will actually, spot correlates to or corresponds to a single molecule. That covers a few pixels. So you're able to have a little wiggle room. You can move the images around to get the best fit. That's what happens in the image processing.
Yeah, when you say wiggle room, so I can go across 2 molecules, it's still not a lot of room there. I mean, the precision is amazing.
Right. It really is. We're at nanometer level precision, which is essentially the wavelength of light that we're using. We're in the 600 nanometer wavelength.
So
region
you
for
have this
imaging.
technology. It can tell you individual fragments of DNA, how they're made or what they're made of, or even more importantly in many cases the RNA, because the DNA stays with you your whole life. The RNA is going to show you the changes that you've gone through. You got this great technology. Where do you go? I mean, it's always great to have scientists do stuff, but now how do you pick a market? What do you do, Liz?
Well, so it's important to note that the technology was originally developed to sequence whole genomes, and it's possible to do that. It's certainly capable of that. But we really believe that the technology, because of the short read and the enhanced accuracy that we're able to get with this technology, we really believe that the sweet spot is in molecule counting and looking at the changes in RNA transcriptome that happen over time. As you said, the DNA by and large doesn't change, and once you know which mutations you have, that tells you something about your risk for cancer, for instance, or another disease, but it doesn't tell you right now whether you have that disease or not, or whether you're at risk for getting it. You know, you're in the early stages and might come down with it soon. The RNA transcriptome tells you in a more real-time way how changes are occurring in your body based on your genetics and based on your interaction with your environment, what you're eating, what you've been exposed to, infections you might have had, many, many different things that may all together come together to lead to you having a cancer. And by measuring with high accuracy over time and beginning to correlate some of the changes that happen in the RNA transcriptome, we can begin to look at patterns that emerge that say, if you have this pattern, you may be in the early stages of diabetes, and even though you don't know it, or you may be in the early stages of heart failure, even though you're not showing symptoms. And we believe that that's a very powerful approach that will really transform the way that medicine is done, enabling doctors to treat early or to begin counseling their patients on dietary and lifestyle changes that might eventually head off, for instance, diabetes before it begins. And so we believe that is one particular application that is very exciting and has many possible uses. We mentioned NIPT before, non-invasive prenatal testing. That's another application because of the accuracy of molecule counting that really differentiates us from what's out there. Now, certainly many people are doing NIPT with other platforms, but our ability to count molecules without bias, that's due to doing a PCR amplification step, really enables us to look earlier in gestation, we believe, and also in certain cases where that fetal fraction that we were talking about, the amount of DNA that's coming from the fetus compared to the amount of the mother's DNA, where that's very low and difficult to detect, and we believe we'll be able to detect it much earlier.
Now, when you think about doing a whole genome, 3 billion bases.
Right.
If I missed a few here and there, you know, what's a few bases amongst friends? But when I think of a short read, you know, if I'm looking at a segment that's 50 bases long, if I miss 2 or 3, 4 out of 3 billion, statistically negligible. 4 out of 30, you know, you're over 10% error rate. I mean, is that the beauty of the technology, that you really aren't losing or missing the bases that are significant?
Well, yeah, you aren't missing them because essentially with the error rate associated with our platform now, it's about on average, one base of that read, which is 30 or so bases, will have an error of some type. Now, what's interesting is because we're a single molecule, the primary error mode is a deletion, where you miss the spot. It doesn't mean, though, that you didn't properly extend it or sequence it. Right. It's simply you blinked and you missed it.
The light bulb on one of them didn't turn on.
Mm-hmm.
So you have, in the past, we've had to design algorithms that can compensate, and it works quite well. So really, there's not really a huge negative effect. The interesting part, though, is that's why we can relate this. People at home are probably thinking, well, why is there so much? If there's 3 billion pieces of DNA, how can you do any of this with your technology that's only producing 30 base reads? Well, it's because when you sequence the samples, you're actually not sequencing very long molecules. Almost every platform that's available chops up the DNA into smaller pieces. And in fact, the human body also goes through that same process, same process in what's called apoptosis. So that's utilized as we degrade DNA from our cells. When there's cell death, essentially the DNA that's very long, that's stored in the nucleus, gets extracted, broken down, chopped up into small pieces. And that's essentially what we use for our blood-based applications. So it's very interesting. And as it applies in the RNA space, you have these long pieces of DNA out there that program all of us. For the RNA, there's this whole new field called non-coding RNAs. Some of the discoveries were made around the corner at UMass Medical. There's a Nobel laureate there that actually helped to do some of this early work. And what we're seeing now is there's not only small microRNAs and short RNAs that have impacts in cells and function, but there's also very long non-coding RNAs that actually bind to genomic DNA and act as repressors. So those molecules, in essence, block the transcription. They block the copying of DNA into RNA, and RNA is then used to create proteins, which are the active molecules in our body. And so by reading the RNA, by doing exactly what Liz said, we can determine epigenetic effects, and this is really a nice topic area for us going forward. We'd like to make it clear to everyone, so maybe we break up the discussion a little bit, but in terms of where we are now, we're able to allow researchers to investigate these phenomena. And we think the technology that we have at the company is very valuable just for that reason.
One other point to make about the error rate is that even though we have some degree of errors, as every other sequencing platform does, because we're able to sequence so many molecules in parallel, we are in essence sequencing many copies of each segment. So when you align those all with the sequence that you already know, or even if you do a de novo alignment, which means that you just start attaching them together almost like pieces of a puzzle until you get one long sequence, what you'll find is that you can tell which ones were errors in sequencing compared to which ones are in every copy that you've made, and those are actual point mutations.
Well, I mean, from what you're saying, let's just keep the numbers simple. I have 1,000 copies, and in the 12th place, the 12th slot, a couple of them don't have the A that all the others, that 994 do. Well, then you can use bioinformatics to say, obviously, that was an instrument oops, because it's randomly missing here or there. And the way you talk about laying layers, I can figure out which ones were just, hey, the light bulb didn't light up.
Exactly.
Or I blinked.
And the key there is that it's random. So these errors are distributed randomly throughout the sequences, and that's much easier to correct for than a bias that can be generated in other ways.
And the bias, if I understand right, when I have less sensitive technologies being used, I have to make many copies. So I take one and I start copying it. Well, if there's 100, and I know there's thousands, but if there's 100 of these molecules, some are going to be a lot stronger than others. And as I start copying, I'm going to copy the strong ones more often. They're not going to fall apart as easy. Right. So the little guys that may be the important ones get washed out. So in some technologies, I may not even notice them. And that may be the key to early detection. Because as you say, when I think of the market, we hear all the time, if you catch certain indications, certain cancers, catching it earlier. When we had our show on breast care, it was obvious that women should take it serious. I don't understand this mammogram issue of cutting back, because they're saying it's not statistically relevant. Well, if it's my daughter, my wife, my mother, I don't want to hear about the other 900 that doesn't matter. Right. To me, it is statistically relevant. Right. It's like Dr. Jones, I hate to tell you, but good news is 99% of the people will survive this operation. Bad news is you're the 1%. You sit there and say, catching anything earlier is got to at least allow you to explore more options. Yeah. So you go in, you've got this technology, you start looking at the market. Where does earlier, more accurate information help? Then what do? You know where you want to go. How do you pick a team to get you there?
So that's one of the things I like the most about a small startup environment is picking a team to accomplish a goal. And what you need to look at is covering all of the different disciplines and all of the different capabilities that are necessary in a biotech company or medical device company. Obviously, you start out with the technical pieces first, and that's exactly how SeqLL started. Initially very heavily focused on the engineering, taking the technology that existed and making improvements to fix some of the very well-known issues that had come across as customers were using it out in the field, as the engineers and scientists at Helicos had been using it themselves. And so that core engineering team spent nearly 3 years, actually, making improvements on that. Now that we've gotten to the stage where a lot of those problems are solved, we're starting to build up the team that's necessary to start thinking about, all right, what applications can we bring this technology to bear on, going out and touching base with people out in the field. Who would use this technology? Where are the pain points that they're currently experiencing with the technologies that are out there right now? And that requires people that are very comfortable going out and talking about the technology with the people that are using it, the clinicians and the researchers and everything. And so we've been developing team members that can do that aspect of the work. We also believe that it's really important to offer a service, sequencing as a service, and so we've got a small team in the lab that continues to run samples for researchers out in the community to continue to do R&D. So some of those samples, we simply sequence them using our technology and provide data back to researchers, and we get feedback from them on how things are working and what they would like to see. We also do some R&D types of collaborations with people to continue developing new applications using the technology, and that's a mechanism for getting people comfortable with SeqLL and what we can offer. And so that there's an interest potentially down the line in purchasing instruments to run these types of analyses on their own.
But now, one of the things I hear across the board in any of the biotechs, the pharma, attracting and keeping people, the good people, because you can always attract and keep the not so optimal people, is
the
one
ones
of the
that tend to
biggest.
stay put the longest.
I was going to say, yeah, the ones you want to leave won't. But is that something that you see as one of the biggest challenges that young companies face?
It is absolutely a challenge because in the early startup phase, you tend to be cash strapped. You don't have a lot of resources. You'll often have a core team of people that are very dedicated, and that was the case at SeqLL and still is. And those are people that are willing to come in early because they're excited about it and they believe in the technology, and they're willing to work for less than they might be able to get at another company. But that phase lasts for a particular amount of time, and after a while, they start seeing some of the bright, shiny objects off in other companies, and begin potentially being attracted
And a lot of
away.
them have these unreasonable offspring who want to eat every day, want clothes.
Exactly.
Want to go to college and
Wanna go to
save some
college.
tuition funds. Yeah, right.
I mean, we saw a change, I want to say back 7-8 years ago, where, and this sounds horrible, but the price of a PhD went up over 20%. All of a sudden you turned around,
The price to hire one.
the price to hire and keep one.
Right.
You know, because you had all the big pharmaceutical companies saying, hey, this is a great area of the country because of the Harvards, the MIT. I mean, you think of all the wonderful schools we have and all the opportunities. A lot of these people tend to stay around. Well, when all of a sudden the Novartises, the Mercks, the Amgens declare they're putting their toe in the water.
Right.
Well, their toe was bigger than my torso. You know, when you started saying, okay, we're going to hire 200 or 300 technical people. Well, all of a sudden the price of poker, the ante went way up. We saw what we were paying for PhDs go up $20,000, $25,000 over a very short period of time. Because like you say, even though, you know, the love of the technology may be there, but all of a sudden little Sally has to go to college at 60 grand a year.
Yep, absolutely. It's a double-edged sword with being in a biotech hub like we are. There is access to some really phenomenal talent, but you're competing for it with big players that have a lot more money than you do. And there are ways you can compensate for that. There's certainly greater flexibility generally in a biotech startup. There's the ability to have equity in the company in a much larger way than you might otherwise have in a large pharma or a larger biotech. So that tends to attract a type of people that that are attracted.
Yeah, and
to
I
those
always
types of
love
things.
it. I mean, Dan and his lab crew, they have the flexibility all they want, as long as they come in on Saturdays, on Sundays, stay till 8 o'clock at night.
Yeah.
Hey, do you
Right.
find that, I mean, it's got to be a culture shock. Somebody coming out of a Pfizer, where they work their 8.101 hours a day, or, it's fixed. They need something to happen, and they've got an army of support people. In startups, we tend to have that delegation mirror. I love the mirror, because that way I can delegate it. You do it, and then I realize who's looking at me, pointing the finger right back.
Very true.
Do you find the culture shock that people have if they haven't been in startups is hard to get over when they come from big pharma?
Well, I think it's definitely the case. In our current situation, we haven't dealt with it too much because most of the people, it's as part of a hiring strategy, you want to bring on people who can function in the environment you're creating. And that's what we had tried to do from the beginning. It doesn't always work perfectly, but you like to know people on a personal level if you can. That really helps because you're investing in a good person who's going to do what they can to make the company succeed. And it makes the working environment that much just more accessible and enjoyable for everyone. For us, it is a David versus Goliath type of situation where very small company compared to all these these massive companies, Amgens and Biogens and billion-dollar companies that have moved into Cambridge. And that Kendall Square area has changed so much in the last few years. We were just there yesterday morning, and it's amazing to me how many probably billions of dollars have been sunk into that ecosystem in the last 5 years alone. And for us, we people at home will, I'm sure, relate. We moved up to Woburn. We went to the cheap district. We tried to save some money, and we ripped up the carpeting ourselves. We painted the walls ourselves. We did the work we could. Thankfully, we didn't do the electrician work. We didn't do the plumbing work. We may have tried, but we just didn't have that confidence, and the city definitely wouldn't have allowed us. But
I was going to say, there's that permitting, that little pesky
Pesky.
inspector
All right.
that turns around and says, you had a chemist run the wires?
Yep, you learn a lot doing that, and I think that's it only helps you in all areas of your life. So that's why we encourage people to if you have an idea, to push it, to see what you can do with it, because you'll never know by sticking to the 9 to 5, and you may have better security, but really there's a window for every company, for every technology. Even Apple reported lower earnings.
For the first
who
time.
thought that was possible recently? So there's a window for every technology,
But even,
and we have to try to hire to hit it.
The old impression that if I work for a big pharma, I'm in for life.
no
It's
longer the
total
case.
stability. And then people started realizing that, okay, this company has decided they're getting out of the cardiac market.
Right.
So all of a sudden,
Or they just got
3,000
acquired,
people
or-
or they got acquired. And most of that research now is going to California or going overseas.
Right.
Or they just have a duplication in management and have to let some people go. And there are lots and lots of reasons why large companies are not as stable as their reputation would.
Yeah, and when they let go, now I'm competing with, if I'm a bench chemist and they let 500 of us or 200 of us go, all specializing in one field,
Right.
boy, that's going to make it hard in the short term for the community to absorb all these people that work on cardiac research, all these people that work on, a specific area.
True, but in the same vein, you have, we moved to Woburn for a specific reason. I looked at areas out in Milford as well, off the 495 corridor here. It just wasn't as close to the people I knew that had the technical knowledge we needed, but it's not that we couldn't have come out here. So by staying around, by keeping a startup in this general area, you know you have access to an educated workforce, a trained and technical workforce if you need that. And so I think that's very important to have that development continue here, in this area. So there's a lot we can do around the world, hopefully, in the next few years. But I still think our core is always going to be in this area, because you take it for granted, but you have so many highly trained and intelligent people here that can really help. And I think at a startup, the bar gets set a little higher. You're thinking, oh, someone has to have experience, they have to be proficient in what they're doing, but also be willing to put in that extra effort. It's not a 9 to 5.
But you also can look at it that somebody coming to a startup is going to get experience in a broader way. Because if you're working at a 500-person facility, you have your niche.
Right.
At a startup, you can be involved in all sorts of things very quickly that early on in your career, or even late in your career, you would never have the option or the opportunity to really play and learn.
And I think that actually gets to one point I wanted to make, which is that I have noticed, as I've been involved in different startups and also talked to many people in pharma and larger companies, it seems to me that there's really quite a divide between the two workforces, between those two pieces. And it's actually quite difficult to go from a startup environment and to land a job in a pharma environment, and vice versa, because as Dan said, we're looking for people that can function in a very small team, wear many hats, are motivated and self-starters, and can just take something and run with it, but still be very good at communicating, because it's such a small, tight-knit community. There may be some of those people in pharma, but there are also very many people that are very used to having their job and producing their work, and it's just a very different way of functioning. And vice versa. I think when pharma looks at someone that's been mostly in biotech, in that teamwork environment, they think, do you really have are you going to be happy in our environment, where you're going to have less control, less influence outside of your own job? And I think in many cases,
you're going to be much higher regulatory burden to deal with.
Right.
I remember when we were in Unison, we had a fellow, greatest guy in the world, incredible researcher. But even at a startup, we found out he was coming into our GMP suites at 2 in the morning, because it was easier to go around the pass-throughs than to go through them, and it saved him. I couldn't argue with him. It does save 30, 40 minutes. Of course, it invalidated every safety, issue of environmental concern.
Right.
And we had to explain to him that although he got it done quicker, and it should be the same, should be isn't good enough if you're going to inject it into somebody.
Right.
So all of a sudden the batches that, quote, were done faster became research grade only for animal research and not even for trials. But it was a different mentality. You could see he wanted to help. He would get up and come in at 2 in the morning. And that was to him, why 2 in the morning? Well, nobody's there. Okay, why do you care that nobody's there? Because then I don't have to follow the rules. It's like, oh. But you can see it where, there's a person who came out of academia, in the university, where it is the Wild West. It's cutting edge. There is no real compliance to worry about. They're just doing proof of concept. And you try and put them into a regulated environment, and it's me speaking Swahili.
Right.
I just don't understand the language.
Yep.
Now, one of the things comes up all the time. We got kids in college, and it's the old adage, you won't hire me without experience. How do I get experience if you won't hire me? What would you say to some of the young students? How do they get out and reach people? Because their goal is to get presidents, CEOs' attention. How would you advise them to?
Well, I think there are a lot of networking events that are available in the Boston area that are really great places for young students to come and rub elbows with a lot of the people working in the field, so that's one thing. There are also internship programs that we would highly recommend. The Mass Life Sciences Center offers one, and we're a member of that, and take advantage of that to bring interns in from college for the summer, and give them some of that experience that you can only get in a biotech company, doing things that you've never done before, and being given the training and experience and exposure and all of that. So those are two I would highly recommend.
But Dan, you look at, we post an internship. Okay, and it's great, Mass Life Science, God bless them, they're paying for it. It's a wonderful thing. So they really do encourage companies like us to take on interns. Great program. The minute you post it, you get 50 to 100 resumes. They all look like the same colored sheep. And you look out in the pasture, it's a bunch of 200 white sheep. How does sheep number 38 get your attention? What do they do so you'll look at them?
Well, I think it depends where you're targeting for that individual. If they have a specific career in mind, then they're going to want to target those opportunities. For an instance like ours, where you're looking at a startup, I'd want to see capabilities in a few different areas, or at least a willingness. And we've spoken to students in the past who had taken a diverse curriculum from economics to literature, chemistry, and molecular biology. I think that's really, that shows kind of a willingness, and maybe that's just my bias, having more of a liberal arts background than hardcore technical. But at the same time, you really, I don't think there's a secret sauce. It's sort of like applying to the colleges now. They're so competitive. You have to cast a wide net with what you're doing. And overall, it makes you a better person in the long run for knowing these things. It makes the real world more interesting, knowing how things work, knowing what the rationale is behind why they put an exit ramp somewhere, right? Or just a bad example, but why somebody has the lighting at a certain level, or a marketing sign in a specific location. There's science behind all these decisions. And so exploring that, there's a lot of avenues you can go to improve your life and your understanding, and that's what I would encourage students to do.
Well, I mean, Liz brings up networking. I'm amazed at how many people don't understand. I mean, even at your level, okay, there's not much more you can do. You've made it to CEO. Okay, where's the growth in the company? Well, there's not going to be a lot of positions there. But yet you belong to a Women in Science Executives.
Yep, I belong to that. I belong to the Healthcare Business Women's Association and Women in Bio, and a number of different organizations. And I try to get out to a couple of networking events a week if I can, at least once a week. And early on, I must say, when I first started at SeqLL, my first job out of academics, I knew nothing at all about networking, and I was terrified of it, and I had no network at all outside of the company. And when I became CEO there, I suddenly realized, boy, I have to go out there and start trying to raise money. I need to have contacts. I need to know who to hire. I need to know where to go for services. And I had to force myself to go out to these events and start talking to people. And early on, I focused on trying to sell myself and trying to get as many business cards as I could, and trying to find some deal at each event. And I, through some coaching and some really excellent advice from a number of people, I learned that it's really not about that. It's about being present over time, meeting people, getting to know who they are, understanding what they need, and then keeping them in mind as you go through your day-to-day business, and referring people to them, or sending them information that you came across that you think would be of interest to them. And those are the kinds of relationships that end up yielding the best leads in the end, because somebody else, they get to know you better, they know what you're doing, they understand what you might need, and lo and behold, you get an email from someone one day, and it's this fantastic person, or somebody that they think you should meet that could be a fantastic business deal for you. So it's really about being part of the community on a long-term basis and growing those connections more than it is about be networking.
But even in the beginning, when you think about it, you mentioned liberal arts, and I laugh because I have a Bachelor of Arts in chemistry. because Holy Cross will not give out a Bachelor of Science. They find that it's too limiting. You've learned more than just the science. It's like, yes, but when I want to go sell myself and I say I have a BA, a lot of people say, well, no, we want a BS. Yeah, we want that Bachelor of Science.
Does anyone want BS? I don't know.
Yeah, is the BA BS or no? You know, it's funny because I've advised so many students, start with your own internal network. You know, if you went to ABC University, they're all or most of them are very good at keeping track of the alumni, so they can put their arm on them and ask for donations. But if you want to get into biotech, first thing I do, you know, go to Mass Biotech, you know, massbio.org, find the companies that are doing work, figure out which ones are most interest to you, and then I'd immediately find out if any of alumni from my school work there.
Right.
Because, I mean, if one of your, you know, alumni from your school calls and says, could I have 10 minutes of your life sometime just to ask you, how do I get started? I mean, how many people are going to really say no?
Right.
You know, it's hard.
Yeah, I think that's a great point. On a personal level, you know, that's how you really make these connections. And especially in biotech, it seems one person moves, you'll get 3 or 4 people following within a few months or a year or so. And there's kind of packs that migrate. But it stresses the importance of trying to be, you know, well-rounded, good person. Don't take things too personally or get offended if a business or a decision doesn't go your way. Push forward and, you know, make the most of your opportunities. And that's how, you know, we have most of our talent today is because I've tried to do that over the years, and I believe it's paid off to an extent where we have people that are willing to come to a small company and work with us in these times, even though they're probably getting paid less than they would at a Pfizer or a Waters
Probably.
or a large company.
Almost certainly.
Almost certainly, but it's, you know, you don't have to overcomplicate
But we do have a
it.
very incestuous industry. I mean, I'm amazed at how close people stay. You know, you don't see them jumping out to the oil industry or then to a copier company. And, you know, you turn around and some of the people you met 10, 20 years ago, you still run into at conferences. Or like you say, now I go to a new company and, oh, it's Joe. Now you've got a unique challenge. You've got to fund the machine. You know, because initially, as Dan's developing new toys, showing people how to use the new toys, that's all the fun. But you're not always breaking even.
Right.
How do you where do you even start? In 2 minutes or less,
2 minutes.
you sit there and say, okay, you've got to go raise enough money to buy the gasoline while Dan's got the engine running. How do you even start to say, hello, would you like to give me a few million?
Well, we've been very fortunate that we have a very committed investor who has funded the company up until this point and continues to do so. But obviously, that's there are
Not
different
everybody
types.
has a wonderful family like the St. Laurent Institute
Right.
that truly believes in the technology, that truly believes in helping people, and are willing to put their money where their thoughts are. I hate to say where their mouth is, but you know, that family's really helped keep this company going.
Absolutely. Yep.
If you don't have one of those, where do you go?
Well, that's where the networking comes in. I mean, this community is very rich with VC, venture capital groups, with angel investors, with you know, financial institutions, and they're all out there looking for the next big company to fund, the next great idea, the next, you know, excellent management team that they can really get behind and put their money into, and you know, of course, the goals of the company may not be exactly the goals of those investors. They're looking to make you know, a certain return on their investment. But in this community, there are a lot of really savvy investors who've been in biotech for a long time, and they have a lot of experience. It's very rarely only about the money here, and so along with those investments comes a lot of expertise and knowledge, and you know, having those on the board can be very good. it's, you know, being plugged into that community and developing a reputation as someone who's a straight shooter and who, you know, is good at developing teams that can tackle these problems and solve them. The more you get out there and talk about your technology and what your plans are for growing this company, what the end goal is, you know, and just approaching those investors and, you know, sometimes doing it through your network. It's sometimes hard to get one of them to pick up the phone, but the more people you know that have successfully funded companies that could introduce you and say, you know, this is someone really up and coming that's, you know, worth your time to look at it and see what you think. That's really how you approach it, and you just keep doing it until you find someone that believes in what you're doing and is willing to put some skin in the
A lot
game.
of slam doors, huh?
Yeah. Although I would say with this technology, less than with my previous company. This one's an exciting one, and I think people see the opportunity here, given how much money's been invested in it in the
Well, I
past.
can see where such an incestuous community is a dual-edged sword.
Absolutely.
So if you are a straight shooter, if you have a reputation of being, you know, upfront person, well, a lot of people will know. If you're one of those people that, you know, we've all been in meetings where you walk out and you feel like, I need to take a shower. You know, I don't know what it is about this person, but no.
Yep.
Mm-hmm.
I mean, that's got to make it 10 times harder to dig out of that hole.
Well, it gets back to what we were alluding to before, which is those personal relationships. And you know, our investor is here because basically we founded the company. They were a previous customer, and we connected with them and had a good relationship. And when the opportunity came to create this company, you know, we were able to convince them to jump on board. And we've been going for 3 years now, so we're very proud of that. And you know, essentially we broke even in the first full year of operations, which doesn't hurt.
Which is amazing.
Which for a startup is pretty tough. Now you can always have more money. I would not say that, but you know, for each phase, I think the investors, as Liz said, they're extremely savvy around here, and we'll go to them for advice on what to do and what they would recommend. It will only help us to be open to that feedback. But basically, they're, you know,
Now, Liz,
they know you need to raise money at certain stages to accelerate. They understand that.
Eventually, you got to pay the piper. So vision fixed on how you get out of this, or do you have to stay wide open, you know, as an exit strategy?
Oh, exit strategy for the company? Well, you know, this company has a number of different options, I think, in the long run. It's a tool that both pharma companies and, you know, other tools companies would be interested in. The IPO market, although it's closing a little bit, has been very open recently.
So wide open vision right now.
Big future, yeah, exactly. We're open to talking to everyone and anyone about potential
Well,
opportunities.
thank you for coming on.
Thank you
Our
for having
pleasure.
us.
And as always, we try and look at what's driving the community, what's driving the technology. Here we have a wonderful medical center that we all know what kind of technology they bring in, and these are the people that develop the technology. So to our 6 loyal viewers, as always, may God bless. May tomorrow be a better night than tonight. Thanks for joining, and thanks for helping.
Thank you,
Thank
Al.
you.
That's great.
Not too long since I've been home.